Hey, it's Dr. Randy Papke with Chapter 7 Biotechnology. Once again, my voice is a little rough, so hopefully this will go fine and we don't have to worry about it, but I might have to pause this and have a coughing fit. Who knows? So we're talking about biotechnology, and just a few learning objectives here for you. We want to understand the basic tools that are used in biotechnology, explain how biotechnology is used to do all kinds of things, like making pharmaceuticals and treating disease. Preventing disease is kind of cool. And then we're going to kind of get deep into the terms, and finally we're going to identify strengths and weaknesses of gene therapy. We'll also talk about genetically modified organisms and you know, if they're a good thing for society or not. So starting out here with biotechnology, just to define it for you, basically biotechnology is just using any form of technology to modify organisms, and organisms can be multicellular organisms like corn, but it can also be individual cells, especially bacterial cells or yeast cells or something along those lines. And it could also be just transforming or changing the molecules that are part of living things like DNA and RNA and proteins and things along those lines. The basics of biotechnology is that we want to have practical benefits. And sometimes we'll say the word biotech is short for biotechnology, just so you know. The other term that we use a lot is genetic engineering. And genetic engineering is a little different from biotechnology, only in that genetic engineering really does focus on the manipulation of the genetic material. So again, really focusing on adding genes or deleting genes or taking genes from one organism, putting them into another organism. So you can see that genetic engineering is kind of under the umbrella of biotechnology, but it's not only. Biotechnology has other aspects to it as well. So we'll use these techniques in agriculture. We'll talk about how the yields of certain crops has been dramatically increased, and costs of producing that food is actually decreased overall, and how we can like deal with making plants resistant to pests so that more of that food is made and so forth. We actually use biotechnology on humans to help make medicines, and maybe even eventually to change humans. It sounds a little scary, but that is one possible outcome. And then we do use it for forensics. We've already seen a little bit of that, but basically, you know, the idea of how DNA evidence can be used, DNA fingerprinting, and things along those lines. So there's a few tools that we're going to step through here and get to know them pretty well. Chopping is, you know, it says chop, amplify, insert, and grow, right? So chopping just means to cut DNA, and we'll talk a little bit about how you can literally cut DNA out of organisms that you're interested in and put them in other organisms. Amplify, that's this one, that means to basically, you're going to make large quantities of DNA, so we'll talk about how that works. Insert, that's where you put a gene of interest that you got maybe from up here or from up here, and you put it in a new organism. And then grow is where you're just making lots and lots of copies in a different way from how you do it up here. So we'll talk about these different ways. Amplify is slightly different from growing, because with grow, you're putting it in a living thing. Okay, so let's talk about chop. And chop is requires a few steps here. And the first thing that you need restriction enzymes, also known as restriction endonucleases. And that's just a nice word because we know that anything that ends in ASE, like endonuclease, is an enzyme that does something. So we're going to chop out the DNA from a donor organism. And of course, first thing you have to do is find the gene that you want. So we call that the gene of interest right there, the gene of interest. and then you're going to cut that out using restriction enzymes. Now, restriction enzymes have only this one function. They cut DNA into smaller pieces. In fact, these enzymes are found in nature. They are found in bacteria. It's kind of like a bacterial immune system. They protect bacteria from viruses. So if a virus invades a bacteria, then these restriction enzymes chop up that virus, and the virus can't invade it. So it protects it. But the way that we use this is that we cut out in this recognition sequence right here. We kind of do this little zigzag cut at this recognition sequence, and this recognition sequence is almost always a palindrome, which is read forwards and backwards the same. Remember that DNA has complementary strands, so if the top strand is ATCGAT, then the bottom strand is backwards of it, ATCGAT. So that makes this a recognition sequence that is also a DNA palindrome. So snip, snip there, the enzyme goes snip, snip there, and now we have this gene of interest. It's separated from the donor DNA. So that's chop. What do you do with DNA once it's chopped? Well, one thing that you can do is you can amplify it. And amplify simply means to make more of or to make bigger, like an amplifier in music. So it makes the music louder. In this case, it makes more and more copies of DNA. And the technique that we use is called polymerase chain reaction. Remember this, any word that ends in ASE is an enzyme. So we're going to be looking at an enzyme in there that does polymerase. Now, you've seen the word polymerase before with RNA polymerase. This time we're going to be dealing with DNA polymerase. So stand by as I finish doing a little bit of intro here for you, and then we'll get into the nuts and bolts of this. But basically, polymerase chain reaction, or PCR, is going to be happening inside of a machine. And you can take a tiny amount of DNA, maybe even just one that was just cut out using a restriction enzyme, and maybe it's been recovered from a crime scene, and we're going to make lots and lots and lots of copies of it. And so there's a few steps that we have to do. The first step is we have to heat it up. And that heat, what it does is it breaks the hydrogen bonds that hold the bases, those complementary bases, together. So heat will break those apart. Now, inside your bodies, DNA can make copies of itself as well, but it doesn't deal with heat. So we need to break those bonds apart. And the next thing that happens is that we have this enzyme called DNA polymerase. DNA polymerase, that's the enzyme that makes many copies of DNA. Enzymes always do what they say they're going to do. So here's our DNA polymerase. And the DNA polymerase works if the temperature is a little bit lower, so we have to cool it down a little bit. And then we have all these little nucleotides in here. So we bond something called a primer to the DNA, and then the DNA polymerase comes in and just starts putting letter after letter after letter. It just reads through, it just adding these complementary bases down the line. And when it gets to the end, it's done. So instead of having just one copy, we've opened it up, added all these bases, and now we have 2 copies. Notice that these arrows are going in different directions. That's because DNA can only be read in that 5' to 3' direction. So in the end, you start out with 1 copy, you get 2. If you have 2 copies, then you get 4. And if you have 4 copies, then you'll have 8. So you're just going to keep going on, repeating this with heating it up, breaking the hydrogen bonds. The DNA polymerase adds the bases, and then it's cooled down, it adds the bases, and then you repeat. So it's a cycling of heating and cooling. And once it's cooler, the DNA polymerase can do its job. Pretty straightforward. Again, the idea here is to get maybe billions of identical copies of that original DNA. The next thing is to insert. And this kind of insertion can happen in many different types of DNA, but we're going to focus here on taking a gene from a human and putting it into a a bacterial DNA and then putting that bacterial DNA into a bacterium. So there's a couple of different ways we're using the word insert here, just so you know. All right, so we're going to take this gene of interest. Remember the gene of interest? We just cut that out using restriction enzymes. Maybe we've made lots and lots of copies of that, but we've got this from our source. And then we need some DNA from a bacteria. Remember that bacteria DNA is circular, so that's how you know this is a bacterial DNA. Specifically, we're looking at DNA plasmids. Those are the smaller pieces of DNA that are not the chromosomes, so the smaller pieces of DNA that are found in bacteria. So we cut this gene out with a restriction enzyme. We're going to use the exact same restriction enzyme, and we're going to use it to cut the plasmid. And what that does is it allows, remember those bases that were hanging off the end there with the zigzag cut? What's going to allow us to do is to kind of match up the zigzags, the leftover hanging over parts, with the DNA in the plasmid. And so that's going to allow us to insert the DNA into that plasmid, the gene of interest into that plasmid. So the ends match perfectly, and you just have to use a different enzyme to seal all that stuff up, and now it's one new piece of DNA that has a new gene in it. Then we can, here's our second insert. Our first insert was going here. Our second insert is to insert that into a bacterium. Now, why do we want to insert it back into the bacterium? A couple of reasons. Number one, bacteria will copy any DNA that's inside its cell when it goes to go through binary fission or cell division. So we can actually make copies of the DNA inside the bacteria, which the bacteria are doing all the work for us, which is kind of cool and very, very inexpensive to do that. And the other thing is that maybe we really want this gene of interest to do the same thing that it did inside of the organism itself. So maybe this gene of interest here, maybe that gene is the gene that allows to make your body to make insulin. Well, if you do this process, then the gene of interest you take out of the human, for example, is for insulin. You put that into the bacterial plasmid, then put that plasmid into the bacterium, and that bacterium will now make insulin. In fact, this is how a lot of medicines these days are made, by putting genes that we want into bacterium and having the bacterium do that. If you remember that word expression, gene expression, well, the bacterium is expressing, it's going through transcription and translation. And it's going to, and the daughter cells are going to do that too. We can have millions and millions of bacteria doing the exact same thing that that your own body does, making insulin. The last step is to grow a gene library, and gene libraries are interesting. We're not going to deal too much with the idea of gene libraries. I just wanted to kind of put it out there. Your book talks about it a bit. Mostly they're interesting because you can maybe put the gene of interest here, maybe this one's for insulin, or maybe this one here is for a different gene or a third gene, and then you just kind of keep track of, you just grow those all in the library, and you have all of these pieces of DNA that you can just go in and say, oh yeah, that's the gene for insulin, and you can take that back out of the bacterium if you wanted to do that. So basically, it's a way of preserving that original DNA in the gene library. So something that's really new, cutting edge, really, really cutting edge of biotechnology is called CRISPR, and that's how you pronounce that word, C-R-I-S-P-R. The term is CRISPR, and it's short for clustered, regularly interspaced short palindromic repeats, which like literally means nothing to you, I know. So we're going to go with the word CRISPR. And basically what CRISPR does is it allows you to edit DNA with a huge amount of precision, a huge amount of efficiency, and basically you can do this without having to go through a bacteria in the middle. So you can basically expose a living organism, a dog, a human, a horse, a plant, to this CRISPR process, and it will allow you to change any gene in that genome. CRISPR is also naturally occurring, just like with the restriction enzymes. CRISPRs are enzymes that are found in bacteria, and as far as we know, it's also about dealing with viruses, viral DNA, and using that information from the viruses to actually protect the bacteria against future infections. So that's kind of cool that bacteria have figured out a way to prevent themselves from getting infected in the future. And again, this natural thing called CRISPR. So how does CRISPR work? Let's take a close look at this. So we've got this gene editing, and it's actually pretty straightforward, even though it's totally newly discovered in the last 10 years. So let's say that we want to identify this DNA sequence of interest. Like, again, maybe you've got a defective gene, maybe you've got cystic fibrosis, and we're trying to cure cystic fibrosis. So the first thing that we need to make is an RNA guide. So there's your gene of interest. Maybe that's the cystic fibrosis gene. We need to have this RNA guide. And this RNA guide just matches the DNA that you're interested in. Okay, so that's the first thing. Following all those base pairing rules. A's with U's and C's with G's and so forth. So the second step, after you've made this RNA guide to identify the gene of interest, is to take the enzyme and put it in the target cell. We are going to use a plasmid again now. So we've got our RNA sequence, and then we've got something called the Cas9. So there's your RNA, that's our CRISPR RNA, and this is called Cas9, and Cas9 is your enzyme. Cas9 is what's going to be introduced, and that's going to be really important in this process. Okay, so now that we've got our plasmid and our RNA sequence that matches the DNA sequence of interest and our Cas9, we're going to go ahead and put that into a cell. And then once we're in the cell, the RNA is going to lead the Cas9 enzyme to exactly the location where it matches up with the gene of interest. And the Cas9 then cuts the DNA. And what we can do is, in that location, we can insert a new DNA sequence right where that cut was, where the Cas9 did the cutting. And all of a sudden, you've got your new DNA sequence in the old DNA. This is so much easier than the old way of doing this, where it just took lots and lots and lots of steps. And so this is like, imagine again, once again, you have cystic fibrosis. Literally, the potential here is to replace this gene using this RNA sequence and Cas9 system, where this enzyme comes in and cuts it out, and it will then allow you to put in the new DNA. And all of a sudden, if it does this in every cell in your body, or even like 70% of the cells in your body, you basically no longer have cystic fibrosis. It's kind of incredible. It's not been tested very much to date, but it has been used in other non-human species. So we haven't actually cured any diseases yet using CRISPR. So the potential for CRISPR is enormous. You could use CRISPR to genetically modify mosquitoes that contain the malaria disease and prevent that malaria from spreading. Malaria is actually the #1 contagious disease on the planet, and it kills so many people. Imagine that you could just fix the DNA of some of those mosquitoes, and big bang boom, you no longer have any malaria. There are doing human trials for taking CRISPR and altering white blood cells in order for them to be better at fighting cancer. So that's another incredible thing that CRISPR can do. It's just a ton of potential. And then finally, we use it for things that maybe aren't so very important, but I mean to humans themselves directly. But of course, we do love our dogs, and so we've actually used CRISPR to genetically modify dogs to give them as like twice as much muscle as a typical dog. So I'm not sure how useful that one is, but we've done it, you know. Maybe some people really like the idea of that. It's starting to get a little bit though into some questionable, um, yeah, issues. Maybe some ethical issues associated with that. And in fact, that's what I want to talk about next is what are the risks of CRISPR? There's lots of risks with CRISPR, including there are some legal issues here. So like who invented it? Who can profit from it? There's a lot of scientists out there doing this work. So, they can be making a lot of money from it. Who owns it? There are a lot of ethical issues too. We just talked about dogs, and I don't know if that's ethical or not. But they're also talking about doing these kinds of fixes to human embryos or to editing just sperm and eggs. I mean, that's starting to get into Brave New World kind of issues. We're starting to get into science fiction. We are living in the future with this idea of CRISPR, and it's kind of incredibly powerful and also a little scary. Also, and finally, we don't really know what the consequences are of introducing these genes into the natural population. So we talked about how great it would be to prevent malaria from killing millions of people a year, but we don't really have a handle on if those genes will just stay within that population. Think about crossbreeding, the mosquito from one species can crossbreed with another, and it becomes this very, very challenging thing to predict about the future. So good sides, bad sides, lots of ethical issues, lots of legal issues, and we don't really know the consequences of using this. So yeah, not quite sure where to go with that, except for it's powerful and it's a little scary. The next thing that we're going to talk about is some of the stuff that we've been doing in the past. So far, up to now, if we've been talking about biotechnology, we've been talking about some pretty basic techniques, and we're going to talk about how we use some of those ones that I mentioned before, like chop, amplify, and so forth, and in the more old-fashioned way. Of course, it's not completely old-fashioned because it's only been around for, 40, 50 years or so. So genetic engineering is what we focus on, and that we're going to be using this technology to manipulate genomes in ways that don't naturally occur. And we're going to be using specifically recombinant DNA technology. This literally means recombining or combining DNA from two or more sources. We've been doing genetic engineering for a really long time. We've been doing genetic engineering for, 10,000 years. Right? Because we've been breeding populations, and that's breeding of populations is a way of manipulating that genome. This is what ancestral corn used to look like. This is what modern corn looks like. How did we get from here to here? Well, we did it through genetic engineering by breeding. But recombinant DNA technology really speeds up that breeding process. And you could go from here to here in just a few short years if you had the right tools. And that's what we're going to talk about is the right tools. So the first example that we're going to step through is that of golden rice. Golden rice is named golden because it is this very beautiful yellow rice, a golden yellow. And the reason that golden rice was invented is because about 10% of the world's population actually suffers from vitamin A deficiency. And if you don't get enough vitamin A, you can go blind. About 250,000 children, that's 250,000 children every year, basically go blind because they don't have enough vitamin A. So the idea behind inventing this is that if we could just solve these nutritional problems, then children wouldn't go blind, and that's kind of a really beautiful thing. So, and of course, rice is a staple in a lot of diets, including like Asia and Sub-Saharan Africa, Middle East, and that's where a lot of these issues are happening. So for most of us in this country, you actually make vitamin A from eating something that has beta carotene in it. And beta carotene is something found in most plants. Like it's what makes carrots orange, but you can also get beta carotene in green leafy things. It's just, but it's just a smaller part of it. Carrots are orange, beta carotene is an orangey yellow. Okay, so so what they did was they took two genes from daffodils, right? Daffodils are this beautiful yellow. That means they have lots of beta carotene. And they also took a bacterial gene, and the bacterial gene is there for helping with making the beta carotene happen better. So it makes it brighter. It makes it a stronger beta carotene, basically. So so these three genes are put into normal white rice by cutting them out of the daffodil, cutting them out of the bacteria, and inserting them into the rice genome. And now the rice is able to make beta carotene. And when you eat beta carotene, you make vitamin A. So by doing this way, you're able to increase the amount of vitamin A that you make by 25-fold. That's incredible. It allowed people to not go blind, to children not to go blind. That's, again, 250,000 people a year. This is maybe a good thing. So in 2016, a bunch of Nobel laureates signed letters urging countries to stop opposing genetically modified organisms, mainly because of the benefits of golden rice. Like some countries weren't selling golden rice or allowing golden rice to even enter their country, even though it could save children from going blind. So again, you're hearing a little controversy in there as well as the benefits. In the United States, we don't really have rules against genetically modified crops, and so almost everyone in the United States is definitely using genetically modified crops. If you are not eating strictly organic food, I mean strictly organic, then you are eating genetically modified crops in this way that I've just described to you with the inserting of the genes. 86% of all corn in the United States is genetically modified. The clothes you wear, 93% of all cotton is genetically modified, as is 93% of all soybeans. So what kinds of things are this genetically modified things happening? Like how are they being genetically modified? And the basics are two things. They either have insecticides engineered into them, so insecticides like literally things that kill insects built into the crops, or they have herbicide resistance built into them. And again, remember that herbicides are things that that kill plants. So we'll talk about that a little bit more. But let's start out here with getting through insecticide or insect resistance. Okay, so imagine you've got a corn plant here and you got a little insect. Now, these particular insects are ones that are caterpillars. And that's really important because the thing that I'm going to introduce to you literally only kills caterpillars. So if it was a beetle itself that was eating the corn, it wouldn't kill the beetle. It only kills the baby form of it, the caterpillars. Okay, so you got a crop, it's devastated by these caterpillars, it's just eating all of the corn crop, and moth, usually it's moth or butterfly caterpillars that are doing this. So what we've done is we've taken a gene out of a bacterium, and the bacterium that it was taken from is called Bacillus thuringiensis, and that's BT, that's where this BT here comes from, is the name of this bacterium. So Bacillus thuringiensis, or BT. So the gene is called BT, and you take this BT gene out of the bacterium, put it into corn, and now the corn is able to make this crystal, this protein called BT. When a caterpillar eats this BT, when a caterpillar eats the BT, either this in the bacterium or in the corn, the caterpillar dies. It only kills caterpillars. So when you put that back into the ground and you plant the seed and you grow up the plant, the butterfly lays, or the moth lays its eggs on there, and well, the caterpillar tries to eat it, but it dies. So the pros of this, the good side of this, is that you don't have to spray this the whole field with this insecticide, you know, because it's built into every cell of the crop. The downside is it's built into every cell of the crop, so you can't really wash it off. You will also be eating this BT. Luckily, BT does not have an effect on humans because humans are not caterpillars. So we've been able to reduce the amount of spraying that's been going on with regard to insects. The other one, the other major one is herbicide resistance. And what we've got here is a crop of rice, and that rice is being sprayed with herbicides. But before this was done, the rice itself was modified, genetically modified, to not die when it was sprayed with the herbicide. And so this herbicide doesn't kill the main crop, it only kills all the weeds, and well, killing weeds makes the farmers be able to grow more of the crop. So that's the idea here, that we can grow more of the crop. So herbicide resistance. Those are the two main ones, but there are other GMO or plants and animals that we've been making. So let me introduce a few of those to you. The first one is that we have genetically modified salmon to grow faster. And the way that they've done this is they've basically taken promoters. Remember promoters? Promoters were those things that are on the far side of genes, and they allow the RNA polymerase to bind onto them and make the gene. Well, the gene that's been inserted in here is growth hormone. So we've got a promoter for growth hormone that makes the growth hormone make more and more and more growth hormone. And so that gene is switched on. So we've got transcription and translation going on like wild. And so these fish get much bigger. These two fish are the same exact age, but this one keeps growing all year round, whereas this one only grows seasonally. And so we get a bigger fish in a shorter amount of time. Is this bad? Is this somehow bad for you? Well, there are some concerns here, but most of the concerns are about the environmental safety. If these fish get out into the wild, then they might outcompete the natural salmon, and that could really disrupt the ecosystem. And there are the ideas that there could be some sort of allergic reaction, eating fish that have all this growth hormone in them, and maybe even more than that, maybe just eating things that have lots of growth hormone might be a problem. So I don't think that has a lot of weight to it, but I am definitely worried about how fish that grow all the time can disrupt the ecosystem. And it could actually harm wild salmon populations, which are, some of them are endangered. So that could be a real problem. Another example of genetically modified organisms is this lovely chicken right here. That has been genetically modified to not grow feathers. And of course, it looks ridiculous, right? But it turns out that these these featherless birds are easier to grow and less expensive to prepare because you don't have the cost of taking off the feathers. However, unbeknownst to the people who invented these chickens, they're like really vulnerable to mosquitoes and parasites and disease, and they're like super sensitive to sunlight, so they get sunburned. And of course, they also have trouble mating because, well, the female chickens really prefer male chickens with feathers, even if they don't have feathers themselves. So they got some problems to work out. But basically, these featherless chickens do teach us an important lesson about genetically modifying plants and animals. Like, this seems like a good idea in general, but there's some unintended side effects that we definitely have to be careful of. And so we've got to think about these things before we dive into using them kind of more regularly. So I've already kind of hinted a little bit about concerns, and I will continue this conversation in the next video. So stand by as I process this one, and I'll see you in a minute.